I'm Matt De Silva. I'm the CEO and founder of Notable Labs. In October of 2013, I found out from my mom that my dad was having trouble finishing his sentences. And so I flew home and was really shocked and dismayed to find out that my dad was diagnosed with glioblastoma, which is a very deadly form of brain cancer. They sequenced his tumor, and we found out that, not surprisingly, he had a very poor prognosis.
Many patients end up going through the same journey when they're first diagnosed with cancer. They're treated with the same thing, what's called the standard of care. And a number of patients end up failing it. Like most patient advocates and friends and family of cancer patients, Matt ended up diving into the literature, learning as much as he could about glioblastoma as possible, looking at alternative therapies that could treat his father's cancer.
We were surprised to find that there were quite a few different drugs being tested that were not oncology drugs. So these were things like a drug for pinworm or an alcoholism treatment. And so started pitching his doctor on the concept of prescribing those drugs together into a personalized combination therapy for my father. And I found out that that was a strategy that was going to be challenging because he didn't want to turn my dad into a petri dish. And so then the next question was, well, can we start to do that in a lab and find out which drugs might work for my dad? So we built the lab to take cells from patients, apply drugs both by themselves and in combination to look for the ones that most specifically killed the cancer, and give that information to the oncologist so that they could prescribe those therapies directly for patients.
At Notable, we're really about empowering the patient and giving them options and laying out in front of them, these drugs might not work for every leukemia patient, but they work really well against your specific cancer cells. The Labcyte ecosystem is totally indispensable for covering the ground that we need to cover. We use it for two different processes in the lab. One of those is the actual drug addition to the cells. And so for each patient, we're screening hundreds to thousands of different drugs or different combinations of drugs to find what works best for that individual. And so to do that, we have to be very flexible. So each screen is designed for that specific patient in real time. And so our software combined with the Echo allows us to custom design each screen for individual patients. We also use the Echo for antibody staining, which I know is a little bit unique, but it allows our science team to move very, very quickly. So we can custom design the antibody staining panel for every well in our screen. That allows us to run single stain controls, FMOs, different staining panels on the same patient sample. Really anything that we want to do, we can do with the Echo delivering antibodies. And that also allows us to move our assay development very quickly. We can titer antibodies, run experiments that usually would take a scientist an enormous amount of time pipetting into individual flow cytometry tubes. We can do with the touch of a button with the Echo. For certain patients, they have a very low cell count. And so for those patients, we need to stretch the few cells that we do have as far as possible. And so being able to perform an assay in really low volume is the only way that you can do that. So at Notable, we've been able to screen a lot of really low blast count leukemia patients that really no other technology exists that's able to screen those in a high throughput manner. For every drug, we're able to give the doctor a score on how well that drug or that combination of drugs killed the patient's leukemia cells, but also a score on how specifically that drug killed the leukemia cells. And we found that the drugs that target individual patients' leukemias are very different from patient to patient. So for each patient, there's a very different panel of drugs that are actionable.
We have to be urgent because there is likely a patient in a severe state behind those cells. So we want to make sure that we can get results back to the doctor as fast as we can. With the Echo, we're able to essentially load a map file that goes into the Echo that tells them exactly what drugs to shoot from a drug plate into our cell plate. And the Echo just blazes through the entire process. We're able to shoot 2,000 transfers in about 15 minutes. In contrast, if I were to do 2,000 pipetting operations by myself, that would probably take a few hours, if not an entire day. The assay from end to end is about 3 days.
The Echo enables us to save large amounts of drugs in our process, and it also avoids unwanted chemical reactions and the imperfections of traditional liquid handling using pipettes of getting accurate results at very low concentrations. So it's been a huge component of us being able to apply the different combinations directly to the patient's cancer cells. We can give it very complex instructions that would be challenging to program in a traditional liquid handler. So the whole workflow is different. You really rethink the entire process when you have one of these tools at your disposal. So what excites me the most about coming to work every day is that when we are delivering results to clinicians, it's directly has a chance to impact somebody's life. So there's no ambiguity about sort of adding value, right? Which was something that my dad was always very adamant about telling me, was make sure that you do something that adds value.